Hi everyone, welcome to 2015 and Happy New Year. For this week's podcast, the Autism Science Foundation would like to look back at the year of research. The podcast is also available as a blog on the website, and we have included some pictures, so please check it out. As we all know, 2014 was a big year for autism research, and there already actually have been a number of perspectives on the science of 2014. In fact, the Simons Foundation did a beautiful job outlining some of the major scientific findings, the hot topics, the notable new tools that were discovered or developed in 2014, among some interesting quotes and artwork by individuals with autism. Please take a look at their year-end summary on their website, safari.org. In addition, ASF would like to provide additional perspective on what's been accomplished in research and where the science is leading. First, genetic findings made a major impact this year. Resources like the Autism Sequencing Consortium, the Autism Genetic Resource Exchange, the Simon Simplex Collection, and the Autism Genome Program were harnessed together to identify different types of mutations and, more importantly, map them out to see how they all fit together. After decades of identifying small or large mutations in single genes, the scientific field was ready to understand the relationship these genes have to each other and to common pathways of function. The best example of this were two papers published in Nature and Nature Neuroscience that were actually published in the last half of the year. These papers laid out the relationships between the different categories of genes associated with autism. One new category of genes that was added that was not really kind of illustrated until this year was related to chromatin remodeling genes. These are genes that don't control protein synthesis, but actually decide how the DNA is shaped. Because you have enough DNA to reach the moon if it were unfolded and straightened out, the DNA needs to be stuffed into cells and shaped properly. That's where it gets its helix shape. An analogy to this is folded laundry. If the laundry isn't folded properly, clothes wrinkle. Like laundry, if DNA isn't folded properly, it doesn't express proteins in the right way. Changes in gene expression without an actual change in the DNA sequence is called epigenetics. I think you've heard me mention this before. It's an area of research that needs more study in autism. Given findings this year and the role of the environment on epigenetic expression, it's likely to become a research focus in 2015. Another important approach that was advanced this year was the use of sequencing and understanding genetics. Instead of looking at individual mutations of amino acids, this time the entire exome, or the code that directs protein synthesis, was sequenced. Through this method, more genes related to autism were discovered, and the way these mutations were passed on was identified. One of the major contributions from sequencing efforts was the confirmation that genes that are highly expressed in the brain are differentially affected in people with autism. There was a lot of work that went into that discovery. For example, how do we know what a gene that is highly expressed in the brain is? And how do we know what happens in the brains of people with autism? For this, we need brain tissue. In 2014, the Simons Foundation for Autism Research, Autism Speaks, and Autism Science Foundation launched a new awareness and outreach campaign for a new and improved brain tissue collection called the Autism BrainNet. Instead of one collection site, there will be 4, so hopefully there'll be 4 times the numbers of brains. Through brain tissue, we saw a number of important findings that may lead to treatments. For example, it took decades to accumulate a data set large enough to show this, but it was shown that a type of immune cell in the brain called the microglia is turned on in relation to how brain cells that control neurofunction are activated. There's been much research over the years on the role of microglia in the immune system, and this relationship suggests that while microglia may not be the singular cause of autism, it may reflect ongoing abnormal processes in brain function. The role of the immune system in autism is another hot topic for research. In another breakthrough study, a group at Columbia showed that people with autism have an overabundance of dendritic spines. Dendritic spines are the points on the neuron that make contacts with other neurons. They took their study a step further by then studying the dendritic spines of an animal model of tuberous sclerosis. This is a disorder with behavioral similarities to autism. What was remarkable was that when they treated these animals with the drug called rapamycin, it reduced the overabundance of these spines. If this line of research continues to move forward, this may mean that there's a new viable medication for autism symptoms that's linked to brain pathology. What we need is more research and more brain tissue. In addition to just looking at genetics, this year an important study was published confirming the interplay between environmental exposure and an autism gene. While studying genes and environmental exposures independently is important, scientists have agreed that it is the interaction of the two that may be more important. In 2014, 2 published studies from the United States showed an association between high levels of air pollution exposure and increased autism risk. In the third, Heather Volk at University of Southern California showed that air pollution increases risk, but this risk was further elevated when a mutation in an autism gene is present. So, does air pollution cause autism? Maybe not, but it's clearly a risk factor that doesn't help brain development. As high levels of air pollution are associated with so many adverse health outcomes in children and adults, it's hard not to argue that it should be a target of prevention. And it's something that's actionable. Regulations and toxic release have improved air quality in the last 2 decades. And in fact, studies in China have reported changes in health biomarkers and neurodevelopmental markers with improvements in air quality. But most importantly, this finding reveals that more studies need to be done, looking not just at genetics, but gene-environment interactions and multiple risk factors. The role of the environment is especially interesting since environmental factors are known to act epigenetically. That is, they modify gene expression without changing DNA sequence. This is epigenetics, which I mentioned earlier. So now that a gene that modifies DNA expression without changing DNA sequence has been identified as an autism risk gene, there may be a valuable target to study. And who could leave 2014 without talking about the new and much-deserved attention to the male sex bias in autism? As you remember, in October, ASF and Autism Speaks co-organized a meeting to better understand the 4-to-1 male-female autism diagnosis ratio. A link to the podcast from that meeting and the agenda can be found on the website. A number of publications also came out this year showed that females actually had a higher burden of genetic mutations compared to males, indicating something was going on that was protecting females from some of the autism symptoms. If we could figure out what it is, and even if they are protected, could we bottle that? On the other hand, are some females with autism just suffering in silence during childhood only to be misdiagnosed or underdiagnosed? This is an area that really deserves further study, not just for the translational potential, but to make sure that girls and females with autism get better services. Finally, some of the most impactful papers came from those studying the early detection and intervention of autism and intervention across the lifespan. For example, for the first time, a comprehensive behavioral intervention targeted at cognition and communication was delivered to infants as young as 6 months with promising results. Also, a low-cost, easy-to-implement intervention for early motor behaviors showed promise in infants. It's still not clear if these interventions prevent autism emergence or diagnosis, but they do improve certain domains related to autism. Another long-awaited early intervention study in toddlers was published and showed efficacy of an intervention called early social interaction. This intervention showed significant effects on adaptive behavior and autism symptoms compared to those who did not receive the same intervention. What was remarkable is that in this study, parents implemented the interventions at home after being trained. But we know that not all people with autism follow the same pathway of the same early signs and symptoms with the same outcomes. This was shown in the largest study so far of high-risk infants tracking behaviors from 1.5 years of age to the age of diagnosis. Understanding how autism symptoms emerge will lead to specific interventions at early stages to improve skills for all people affected with autism. But the research didn't just focus on infants and toddlers. The science around a behavioral intervention called PRT, or pivotal response training, in children and adolescents showed important advances as well. First, children of parents who were trained in PRT techniques showed improved outcome. PRT was able to reverse the differences in brain activity seen in individuals with autism. It also resulted in improvements in language and adaptive behavior. Because of long waits for clinic-based approaches, interventions that are delivered by parents, caregivers, and teachers need further data behind them and need evidence-based adaptation for delivery. There are so many challenges behind getting caregivers and teachers to implement interventions at home or in the classroom, and figuring out ways in which this can be done should be a priority for public health research. The lives of people with autism are made better through research. Before these interventions are declared effective, they have to be studied in real-life settings. Scientific findings must be discovered and then replicated to ensure that researchers are on the right path. This year's collections of discoveries would not have been possible without these researchers, scientists, and research staff who dedicate their lives to people helping autism, and individuals supporting science through their generous donation of funds. But most importantly, the families participating in research and all the people registering for postmortem brain tissue donation also deserve special kudos. Thank you and have a great 2015. If you'd like to read more about the individual studies that I mentioned in this year-end review, please go to the blog on our website. I've listed them numerically at the end of the blog. Thank you, and talk to you next week.